Heyo, folks, what up, dudes? What's going on, my 7th grade science students? I'm here with Jonathan Rittershold. I'm here with Jonathan Rittershold, and we are going to be doing your class today on today's virtual class. How's it going? How's the audio coming through? Audio's good? Jonathan, you can say yes if you want.
Yes.
And then what about the computer screen? Can you guys see my computer screen? Because the computer screen can see you. Apparently, it's got a bunch of eyes on it. Well, that's kind of creepy. All right. So we're talking about genetics and inheritance, all of that kind of stuff. We talked about Mendel and his peas. And how was that? That wasn't that bad?
Yeah, I like the video that you posted on that quiz. That was cool.
Oh, the which ones? I punched a bunch.
The quiz that was due yesterday.
Oh, okay.
That was a cool video.
Well, with this chapter specifically, it's not that I don't know what I'm talking about, but to be honest with you, I'm not necessarily the best at this stuff. Wet koalas. What are you guys talking about? All right. Anyway, it's not necessarily that I don't know what I'm talking about. It's that lecturing on this can be really confusing, I think. I don't know if you've tried to read this chapter. Have you? Have you read the whole chapter? Like all the way through lesson 3?
Yeah.
Because when I read it, it didn't make any sense to me. Like, I read it a bunch of times, and it didn't make sense to me. But then I watched a bunch of videos, and the key to watching the videos is finding videos with lots and lots of animations. And I don't have the time to build animations for you guys. So when I show you videos with lots of animations where they have the funds to be able to make animations and these guys have the time to do it, then it makes a lot more sense. So I'm going to do my best to lecture on it, but pretty much the way you're going to learn inheritance and DNA and genetics is by watching the videos. So make sure you guys do that stuff. Everyone cool?
Yes.
Yes? All right. All right. So I think we talked a little bit about these things right here. We talked. I'm going to kind of recap lesson 2, and then I'm going to do my best to kind of cover up on lesson 3. Eric, you're downstairs. Why don't you just come upstairs?
Yeah, I'm downstairs in the breezeway.
All right. Anyway, we got the Punnett squares. Okay, the Punnett squares. If this were a capital Y, the capital Y, basically the capital letter can stand for the dominant gene, and then the lowercase letter would stand for the recessive gene. If everything here will be capital Y's, then what would be in all of these boxes?
It would be all dominant.
It would be all capital Y's, right? And if everything in there had capital Y's, then pretty much we have what we call the true breeding plants. That would be the equivalent of, in the Mendel and his peas kind of deal, that would be this picture right here. Capital Y and capital Y makes a bunch of capital Y's. Or all lowercase y's and all lowercase y's make a whole bunch of lowercase y's. Does that make sense? Mm-hmm. But if you mix an all capital Y and an all lowercase y, you'll get a hybrid which will look like the capital Y one, but it won't be the same because it has both capital Y and lowercase y. And so we take these flowers over here, and that's what the Punnett square that you are seeing. I don't know if we're pronouncing it correct. Punnett, pungent, Punnett square. This is the capital, or this is the Punnett square you're seeing. This is one of those purple hybrid flowers, and this is another purple hybrid flowers. Maybe the orange is the male and the purple is the female or something like that. Does that make sense? And then when you mix those guys, capital Y's, you'll notice that this flower right here, this baby flower right here, will be a true bred, actually, a true breeding, what you call it, state, a true bred flower, because it will have 2 capital Y's with a dominant. In fact, we would call that. I got a question. Is that a homogeneous or a heterogeneous? Do you know? You can answer, Jonathan.
Homogeneous?
What
I'm not really sure.
do you think? You guys say it. Come on, everyone.
Homogeneous?
You think it's homogeneous?
Yeah.
Okay. You think it's homogeneous. You are absolutely correct, my friends. This is a homogeneous dominant trait because homogeneous means the same. Right? All right. All right. So I know that you guys are thinking this, and fine, we'll go into it. If you think of a homosexual, which I'm sure that you guys know what it is, right?
Yes.
You're talking about somebody who likes the same gender. Well, that's what a homogeneous thing is. You have two of the same traits, right? Versus a heterogeneous, as you know, in relation to like a heterosexual who likes people of the opposite gender. You have the opposites or different. Maybe hetero would be a better word, not necessarily opposite, but different. Different mixture. Does that make sense to you?
Yes.
So now, if I were to ask you the words, does now you can say, oh, I understand the words, because I'm sure that you guys never heard the word homogeneous and heterogeneous before, but you've heard homosexual and heterosexual before at the age of 13, right?
Yes.
Cool. Okay, so obviously this one is a heterogeneous mixture, right? And it will look just like this one, but it won't be the same. And this one is actually pretty much the same as that one. The two, the top right-hand corner, the bottom left-hand corner. Now, the really interesting one is when you have a white flower or a green bean or some kind of recessive trait popping back out after you have two flowers that look like they only have dominant traits mixing together. But that wasn't true. They were hybrid plants mixing together. Are we cool with this?
Yes.
All right.
Cool.
So this Punnett squares is actually really helpful for you to understand genetics. Let me see, did they talk about this? Okay, all right, so check this out. We end up getting a ratio of 3 to 1. And you can see that directly in the Punnett squares. You got 3 yellows to 1. And that almost always happens, right? And we saw in this big table up here, it was pretty interesting that all of Mendel's experiments almost all came out to 3 to 1. Notice that it wasn't exactly 3 to 1. But the ratio is close to 3 to 1. That's almost like saying if I flip a coin 2 times, I guarantee you that one of those times will be a heads and the other time will be a tails. That's not necessarily true.
There's a 50-50 chance.
Well, not even there's a 50-50. I mean, it is a 50-50 chance, but I can tell you for sure that if I flip a coin 1,000,000 times, then roughly half of them will be heads and half of them will be tails, right? But I can't necessarily say, oh, if I produce 4 of these babies, if these plants produce 4 babies, then Garens bobarens, 3 of them will be yellow and 1 of them will be green. No, they could all be yellow, or they could all be green, or something like that. It just ends up being the luck of the draw. Does that make sense?
Yes.
Just like if I flip a coin heads and I flip it a second time and I get heads, and then I'm going to say, okay, I'm going to flip it a third time, and you're going to be like, well, it's got to be a tails to even it out. I guarantee you, I could bet $100 million that it's tails. And they're like, well, I flip it and it could be heads, it could be tails. The outcome of this next flip wasn't affected by the result of the first 2 flips. Does that make sense? All right, cool.
Because like if you just flip heads the first 2 times, it doesn't necessarily mean it's gonna be heads again or it's gonna be tails.
Right.
It can be
But
either one.
if you did it a bunch of times, like, you know, if you counted a large number of offspring from a particular cross, the overall ratio will be close to what we predicted in the Punnett square. Does that make sense? So, the other way of looking at inheritance is through pedigree. And I always think of pedigree with dogs, but you can actually apply pedigree with humans. It just means, like the traits that you have versus the traits that your brothers and sister have versus the traits that your parents have and your grandparents and within your family. So here's an interesting trait. Like here's this, the white squares is fathers or boys, and circles are girls, which is kind of like in the movie WALL-E. The boxy one was the boy, and the more circular, curvy one was the girl. And then you have the, yeah, I know. And the white, if it's white, it means that they have unattached earlobes versus these offspring. If it's orange, it has an attached earlobe. And that is actually a trait that you could inherit as a genetic, feature. And so apparently the parents they have unattached earlobes, but the children have attached earlobes. I would almost expect, so what does that tell you? Does that tell you that which one is the dominant trait versus the recessive trait? Unattached or attached? Which one is dominant?
Attached.
You are absolutely correct, Jonathan. Ritschkajawald. You can correct me if I got your name wrong. Ritterschold. Got it. In fact, keep doing that. It's going to be funny. Yeah, you are absolutely wrong. I like saying wrong because it's like, yeah, I'm correct. No, you're wrong. All right. Well, here's the thing. Look, the way I'm looking at this is I'm thinking that the unattached earlobe is what is the dominant trait. Because if you look at this one, there are no, karang means wrong, by the way. If you look at this one, obviously the parents have this orange gene in them, but it's obviously not being displayed. So they must have a mixture of the dominant trait and the recessive trait. And since only a few of these offspring have the recessive trait, but the parents don't, it tells me that the orange attached earlobe is the recessive trait. And I asked you, what is the dominant trait? So you would say, oh, it's the unattached earlobe. And looking at your earlobes, they're unattached. In fact, I'm wondering if you go around and find all of the people that you know and ask, see whether or not their earlobes are attached or unattached. And it's not like you have to ask their permission for the survey. You can just look at their ears, I wonder if you go around, I wonder if you would find a 3-to-1 ratio.
Is curling your tongue a trait like this?
Well, I think so, yeah. Curling your tongue, making it into a U, that could be a trait because not everyone can do it. So, I mean, maybe that's also your height. All of the things we have, we inherited from our parents. So, here's the thing, my friends, is Mendel happened to choose choose a plant to study that only depended on two things. You know what I mean? And it almost like simplified his life so he can understand the basics of it. Because when you apply it to humans, no, there's way more complex things. There could be different kinds of dominance and recessiveness and all of that kind of stuff. Our eye color isn't dependent on just two different kind of genes coming in and saying, well, this is the answer to that. You could have, so why did he choose a pea plant? Well, I think
It was simple.
no, because he got lucky. Because he just so happened to choose the one that had only depended on two things. If he chose something else that was a much more complicated thing, like for example, a camellia, which is a plant. It's a flower. It's a pretty flower. Do you see that? This pretty flower. He would get confused. He would have gotten confused. So it all just kind of worked out where he happened to choose the one that, only depended on two things. It was really simple. It could be modeled by Punnett squares, all of that kind of stuff. But not everything can be. Like, for example, we talked about dominant traits and recessive traits, but there could be an incomplete dominance. And it's when the offspring's phenotype is a combination of the parent's phenotype. For example, the white flower of camellia mixed with the red camellia flower gives you a pink camellia flower. Crazy.
White and red mixed to make pink.
Right, and so white and red mix to make pink. And remember at the very beginning of this, or beginning of this chapter, I asked you at the very beginning if you had if you had a gray squirrel or a black squirrel and a white squirrel, will you get a gray squirrel? And you guys all said yes, but I told you that's all wrong. Well, not always wrong. Could have incomplete dominance. You know what I mean? So, or is everyone on the other side? There is a person. There is a person where, like a fly in the room, or what? No. Yeah, no, it's John. It's Jonathan Rittershall. Yeah, he can speak up if he wants. I think it'd be funny to have him.
Thank you.
Have him hang out in the class. He's a cool guy. I like hanging out with him. And you'll be able to ask me questions. Make sure you ask me questions if I'm going too fast or I'm getting too confusing or whatever. All right, so I got a question. What happens if you mix a white cow and a red bull? That white cow will have wings. No, I'm kidding. No. A white cow and a red bull. Will you get a pink cow?
No.
No, I've never seen a pink cow. But I have seen something a little bit different, and that happens with codominance. So this picture with these flowers, that's incomplete dominance, whereas codominance appears when you have both of the phenotypes of the parents interacting. So it doesn't mix. It almost kind of like, here, I'll show you.
Oh yeah, I was gonna.
You got a white cow and a red cow make a red and white cow, but not a reddish white cow or a whitish red cow or whatever. Right? It makes a spotted red white cow. Does that make sense?
Yeah.
So that would be a case of co-dominance. What are you talking about Minecraft for? All right. I must have called 1,000 times. What are you talking about? Called who?
It looks like a red velvet cake.
Yeah, it does look like a red velvet cake. All right.
What?
Is that me? No, that's not my... No, Adele? I don't know who
That's
Adele is.
That's my mom's name.
Oh, okay. All right. So, here, check this out. Mendel and his pea plants basically used two alleles to make the color of the peas or the color of the flowers or everything. But there could be more. Like, for example, there are the blood type. You have 4 different types of blood types.
Like A,
You could have
B's.
type A, type B, type O, or type AB. Got it? Now, if you have type A, like see this I with a little A on top? That doesn't mean I to the 8th power. That means you have one gene that is IA, and you have another gene that is IA, then your type of your blood is going to be type A, excuse me. You get it?
Yes.
But there's a different thing. You could have a type IA and a little I, which is a different allele for blood type, and you would still have type A. That tells me that this IA is actually the dominant trait over this thing that kind of governs what happens. If you have a type IB and a type IB, then obviously you're going to have type B blood type. But you could also have an IB and a little I, and that will still give you a type B blood type. This little I thing is actually a recessive trait. So if you have 2 little I's, then you can, I can take the bus. Oh, okay. All right, cool, Quinn. I keep that in mind. Darn, I could really use some help with the thing, the Science Olympiad. All right. If you have two little eyes, then that will make your type O blood type. Does that make sense?
Yeah.
But if one of those eyes is replaced with one of the IAs or the IBs, that thing will dominate as far as what kind of blood type you have.
Be a type A.
And you have a type A or type B, depending on the thing. But if you have a mixture of IA or IB, which one dominates, the IA or the IB?
Neither. They're both the same. So
They're both the same. So or you can say it. Go for it. I'm sorry.
So they're both the same. IA and IB, they're equal to each other, so they just become AB blood.
Yes, exactly. So the IA and IB alleles are co-dominant, and that's what you just said, right? But they are both dominant over the little I allele. See how it could get much more confusing? I mean, that's just blood type, and there's only 4 different things. What about hair color? There's many different kinds of hair color. What about like skin color or eye color or height? There's plenty amounts of height.
Millions of times.
I am not an English teacher. So what I'm saying here is that Mendel happened to look at only one gene, whereas, like when I'm actually applying this to humans or to animals or whatever, you could have a polygenic inheritance, which basically means multiple genes is used to determine a phenotype. And I think in humans there's actually, to be honest with you, I don't know. I don't know how many there are in humans, but I know that there's more than 2. We're not as simple as pea plants. Got it?
Much more complex.
Yeah, much more complex. Are we cool with that?
Yeah.
So that's chapter 2. Oh, the genes in their environment. I thought this was a really interesting slide.
Kitty.
Sometimes your genes will actually change depending on the environment you're in. Actually, that I say your, but I don't mean your. But sometimes genes might change depending on the environment they're in. For example, if you have more acidic soil, hydrangea plants, which is actually one of my wife's favorites, they grow up in much colder climates. But if your soil is acidic, then your flowers are going to be blue. But if they're more alkaline or more base, then they're actually going to be pink. Crazy, huh? Or, for example, a Siamese cat. Have you seen a cat like this?
Yeah, our neighbor has one downstairs.
Yeah. Really,
Yeah.
Siamese cat? Well, actually, I've only seen a Siamese cat in the Lady and the Tramp movie. I don't know if you know.
Yeah,
if you guys
I watched
remember.
it a lot of times.
Yeah. So, like in the Siamese cat, it's got some interesting color patterns. It actually is its genes have told it to make the cooler parts of its body darker. Because I don't know if you did this experiment in 4th grade, but like dark clothes get hotter than light clothes or
They
whatever.
absorb more heat.
Right. So with the Siamese cat, if that's a cooler part of their body, for example, their ears or their nose or their feet or their butt, then their genes have made it. The environment has decided that those are the cooler parts, and then that makes their fur darker in those areas. They're not like a chameleon where they can change really quickly, but that's the way their genes have made it, so that way they can keep those things warm. I think that's really interesting.
And is it the same what the dark skin colored people out in the sunlight, or like
Maybe it could be a case where where darker skinned people grew up closer to the equator, and fairer skinned people maybe originated further away where there's not as much sun. Yeah, hey, that could be a possibility. I don't. I'll be honest with you, and I'm just going to come clean with you guys. I'm doing my best to teach you guys. I hope that I'm a good teacher, and I think that sometimes I'm a good teacher because I don't know everything. But my confession is I don't know everything. And the reason being is I'm a physicist and an engineer, and this is not necessarily my area of expertise, but I find it fascinating. And technically I'm certified to teach you, so so you deal with it. All right, DNA and genetics. I'll be honest with you, when I read this chapter like 6 times, it still confused the heck out of me. I'm going to do my best to explain it to you, and we'll go through it. Sound good? All right. So, This part is talking about what the DNA looks like. Now, the DNA is an organism's genetic material. It's deoxyribonucleic acid. Got it? And this is basically, I think of this as the cookbook of instructions for your body. You know, your body needs to make new things, and so it always has to reference this cookbook. And it's not wound up in a long string because that would take up too much room. What it is they coiled all of this stuff up, kind of like a telephone cord. But then again, you kids nowadays don't understand what a telephone cord is.
Because it's wireless. We usually have wireless phones nowadays.
Yeah, I know, because you guys are cell phone. Yeah, it doesn't look like a cell phone. A telephone cord.
More modern day.
Gosh, what is that shape that you guys would know? Do you guys know that shape? I don't know.
Spiral.
It's like a spiral
I just
staircase.
went to the park before I came here, so I saw one of those things that you climb on like that.
Yeah, they're at the park and whatever. Yeah, no, I mean my telephone cord has that shape to it, right? But that's because I'm old and old school, so I'm old. All right. Anyway, the reason why is so it can really be in a nice compact form and be put together so you can fit a lot of stuff inside there. All right. It's a complex molecule. So here's what I want to tell you guys. There are 4 different types of nucleotides. A nucleotide is adenine, cytosine, thymine, and guanine. So adenine is A, cytosine is C, thymine is T, and guanine is G. Got it? Now, you will always find, when do we finish? I'm thinking we finish like 12:10. This is going to be kind of a long chapter. All right. You will always find the A's mixing with the T's. Got it? And you will always find the C's mixing with the G's. You'll never find a C with an A. You'll never find a cytosine and a adenine nucleotide mixing together. We get it?
And the S doesn't mix with anything
okay.
else.
Now, what the S is, these are sugar phosphates that kind of make up the... If we were thinking of this as the cookbook, then these nucleotides are the letters inside of the cookbook. And you need a few of these letters to make up words in the cookbook. And then I kind of think of these sugar phosphates as the covers. They're like the walls that keep everything in. So that's what makes up your DNA molecule, is it's kind of like bound by the covers of the book. And the covers of the book are made of sugar.
Oh, so the S is the blue stuff on the outside and all this.
Yeah, the S is the blue stuff on the outside, and all of these colorful things, these are A, G, S, or T. Or, I'm sorry, A, C, T, or G, depending on what order you have it in. Does that make sense? Now, I told you guys a little lie, and that's that the A's and T's always bound together. In the next section, we're going to actually see the A's binding together with a U, a brand new thing. But let's not worry about that for now. Let's just think A goes with T, C goes with G. Are we cool with that?
Cool.
A course of lies. He's telling us lies. All right, so here's what happens. You know how your cells duplicate?
Split apart?
They split apart,
Yeah.
right? They go through mitosis and meiosis, and they basically split apart, right? Well, your DNA needs to constantly replicate itself. It needs to be split apart and build and duplicate itself. Because, if your cells are splitting apart, you better have equal amounts of DNA in both of them. You know what I mean? So here's how the DNA does it. I think it's really interesting. First, there's this thing called a heliocase. And that kind of goes through like a scissors. And it cuts apart this DNA strand. So it's almost like you're just cutting it straight down the middle. You guys understand? You're cutting the book in half?
Yeah.
All right, I got a question. what does a 15-year-old teenage boy and a heliocase have in common? All right, this is kind of a bad joke. I'm sorry.
Same DNA. I don't know.
They both want to unzip jeans. Ha!
Oh.
Yeah, that's a bad joke. I can't believe I got that on recording. No, no. Like jeans is in like
DNA.
yeah. That was a little dirty. I'm sorry. All right. So you have these two. The heliocase kind of comes through, and it's almost like it cuts the genes in half. Now, what happens is you have these extra nucleotides hanging around, right? There's just an excess amount, and then so let's see. What was the? All right. So the green nucleotides has an A attached to this thing right here, right? And what was it attached to? It was attached to a T, which is purple on this side. Just like the G nucleotide was attached to the C. So the body says, hey, there's an extra A floating around here. So I need to go and find a T, and it goes and gets a T, and it links up that T to the A. What's up?
Leave this here.
Yeah, sure. It links up that T to the A, and then this T to the A, and this C to the G, and it basically, all right, so the DNA strand separates into a nitrogen bases are exposed, so basically the heliocase cuts this thing in half, and then the nucleotides move into place to form new nitrogen-based pairs, and then two identical strands, basically they put on a new cover on one of these,
So you just
and
cut
then
it
you
apart and split it, and then it becomes two new DNA.
right,
6
exactly,
cells.
yeah, it becomes two exact replicas of this thing. Does that make sense?
Yeah, they cut everything in half.
After they cut it in half. Oh, come on, guys. I mean, I know I threw it before, but... Oh, yeah, hey. All right, well, yeah. Okay. So, like, this one was attached to an A. It's a T, right? So, the body is going to do what? It's going to take which one of these to attach to that piece right there?
Take the blue one.
The green one-ish,
Green.
greenish one. Yeah.
The A.
It's going to take the A and attach that to there because it was attached to an A. So the body says, hey, there needs to be an A here. And then so now when you have this thing kind of filling in all these gaps, it's making two separate DNA. So you have one DNA here. It got cut in half, got built up back together, and then got the book cover put back on top of it, and it's now here. Does that make sense?
Yes.
Did I explain that better than the book did?
Yes.
'Cause it was,
Much better than the book.
I was reading the book. I was like, I'm so confused. All right, I could make you guys do this lab, and it actually looks pretty good. I would actually do this if you guys have small paper clips and large paper clips and pipe cleaners at home. I might make you guys do it in class, but it's a good one. It's a good one. All right. Making proteins. Now, this I think is interesting. One of the things I need to tell you guys is that, like, you would think that these genes and DNA are super efficient and super important and all that kind of stuff, and it is. They are. They're very important. But I think it's really surprising to learn that most of your DNA doesn't actually do anything.
Just there's a
97% of human chromosomes don't form any genes whatsoever. We actually don't know what there are. We call it junk DNA. I suspect that there's no such thing as junk DNA. I suspect that there's a good reason for them. We're just not smart enough yet to figure it out. But for now, we know that as junk DNA. Crazy, huh? And that's way more than half. That's almost all of your DNA is not actually useful.
97%.
97%, only 3% of it is actually what goes into coding, you. That's crazy. So, the reason why I say that is because I think that, like, honestly, if you guys are young scientists, start learning this stuff, and maybe you guys can be the one figuring out what the junk DNA is actually not junk. I think it's interesting.
Hallelujah.
Yeah, that'd be cool. All right, so there's this other thing that's called RNA. RNA can be really confusing. Let me try to explain it to you the best I can. All right? I'm going to use a lot of analogies on this one. RNA is used for making proteins. Now, here's kind of what happens. Actually,
What does RNA stand?
one second. Let me see if there's another teacher out there. Here, can you go see if Celeste wants to hang out with us? Hey, Celeste, you want to come and hang out with us real quick while I explain RNA?
Yeah.
Are you guys going to start up in like 10 minutes with the... Are you going to start up with the 7th graders or the 4th graders in 10 minutes?
About 10 minutes.
All right, have a seat or have a stand. You can.
Okay.
Okay, so I have with me a true expert on this. We've got the physicist trying to teach biology to people, and I'm right here standing next to me is a biologist. So I'm going to completely open myself up to complete embarrassment and invite Miss Celeste Yee in here with us, and I'm going to talk about RNA. All right, so RNA stands for ribonucleic acid, and it's basically it's different from DNA, but it's super important. Its job is to make proteins because that's what DNA does. DNA doesn't make proteins, but what DNA is it provides the instruction manual, like we said, the cookbook for it, right? And Miss Yee, you can always chime in
Yep.
or even say you're completely wrong.
Sounds pretty good so far.
Okay, so the RNA the DNA is like a cookbook. Now the RNA is basically the cooks that use the cookbook to make proteins. Because proteins is what you need to survive, and you need to have certain kinds of proteins in your body, and the kinds of proteins in your body depend on the DNA that you have.
Yep.
Haha, I'm getting good at this.
Yeah.
All right, so your DNA is hidden. Where is your DNA hidden? It's inside of the nucleus, right?
Yep.
All right, it's inside the nucleus of the cell. So there's 3 different types of RNA. One is called the mRNA. I'm going to call that the messenger RNA, and I think I just so happen to get lucky enough to actually think that it is called the messenger RNA.
Yeah.
And what this guy's job is to do is you can't take the cookbook out of the nucleus. You guys remember the cell that we talked about before? It has a nucleus in the middle. You can't take the DNA out of the nucleus. That's not allowed. So what the messenger's RNA, his job is to do, is he goes into the nucleus and he copies part of the DNA. He kind of copies, he cuts the DNA in half, and he'll leave with half of the DNA, basically. And this is the case where we attach a U to the A, and I don't really know why.
Nobody really knows.
Nobody really knows why. Even the biologist doesn't know why. So I don't feel so bad now. All right. So what happens is he kind of cuts the DNA in half, and then so like let's say he's copying this bottom strand right here. Okay. So then he says, all right, there's a C. I'm going to attach a G, and here's a T. I'm going to attach an A, and here's an A. I'm going to attach a U. Again, we don't know why. Here's a C, I'm going to attach, or a G, I'm going to attach a C, and he goes so forth, and then he comes out with his copied page of the cookbook. So you can't leave with the cookbook, the secret cookbook. All of Granny's recipes are in there. You cannot leave the vault, which is the nucleus, with the cookbook, but you can go in and copy pages off of it. Does that make sense?
So, and the RNA is just one strand, it's not 2 strands.
RNA is one strand, and it's actually the process of copying it is called transcription. In fact, I think it tells you, RNA is a type of nucleic acid that carries the code for making proteins from the nucleus to the cytoplasm. Got it? DNA is double-stranded, but RNA is single-stranded. That's where I read that, and I was like, oh, okay, yeah, it's just basically half the information. Does this make sense? Kind of.
Thumbs up.
That's not a thumbs up. That's I don't know, kind of. Okay, all right. You know what? I'll be honest, it kind of makes sense to me too. All right, so here's what happens. Got it? The mRNA is this string of information. And in order to synthesize, and what I mean by when I say the word synthesize, I mean build, because I need to explain to you the words that I'm using. When I say synthesize, I'm building proteins. And in order to do this, remember, this is our cookbook. This is a copied page off our cookbook. And these are letters in the cookbook. Now, all of the words in our cookbook are 3 letters long. They're like cookbooks for 4-year-olds where they get to first learn how to read, and they're all 3 letters long.
If,
Right. No.
what?
But, can, cat, dog, right? But and also there's only 4 letters in their alphabet. There's the A, C, G, and U. Okay? So, what happens is the ribosome, which is part of your tRNA, this guy comes along and reads that 3-letter words, and it says, oh, the 3-letter word I'm going to see is a UAC. Got it? And so I'm going to go find a protein that will match up with that 3-letter word. So what matches up with the UAC?
T, G, and A again?
No, I think it's ATG, right? Because the U goes with the A. In ribosome world, the U goes with the A goes with the T, and the C goes with the G. So it'd be an ATG kind of deal. Does that make sense? Apparently your body's really good at this.
Yup.
And we suck at it. But I'm just saying, I don't know how to do this. But your body's really good at this. And I think that's amazing the way our bodies are built in that way.
Yeah.
Do I get an A+? Why do you get an A+? All right, anyway. So anyway, your ribosome basically goes along this line and reads all of this stuff and spits out all of these proteins that it's building based on this information that it's receiving from the mRNA. And the mRNA was the thief that went into the security vault and got that information from the DNA that was locked inside of the nucleus. So that tRNA is like the cook that reads the information from the thief. Does that make sense? And the rRNA, I have no idea what it does, but he's another cook. He's like a sous chef. All right, he's also important in the process somehow, and I don't know what's going on, but he's there. Yeah, you need 2 cooks in the kitchen.
Especially when you got a busy restaurant.
Yeah, there's a busy restaurant. He's got to do a bunch of this stuff. So, all right,
Yep.
I explained it the best way I possibly could. I'll explain mutations in just a second, but let me take this opportunity to humble myself and shut up while the real biologist corrects the things I said, or maybe even provides interesting insight into what we are trying to learn.
That is perfect.
I did a perfect job.
Yeah.
Nothing to add?
Nothing to add. A couple of the terms, I guess, like the those words that the codon or the cooks read off of the RNA, those are called codons. So
All right.
each word is called a codon.
Those 3-letter words that
Yep.
we're talking about. Cat, dog. In this case, it's AUC, awk, and tag. You know, they have a tag word
Yep.
in there. So there's only 4 letters they could use.
Right?
The only 2 words you can spell is probably cat and tag.
Right, cat and tag.
Or gat.
Gat.
Gag.
At. But
At.
you could. So that's Mr. Dolly explained transcription. So that's going from the vault, right, and then transcribing your pages of information out to the outside of the cell. And when your messenger RNA hits the ribosome, which is your rRNA and the tRNA sous chef combination, then that's called translation. So your tRNA translates what's written on the mRNA, and then that's what spits out your protein.
All right, here's my question. All right, and I'm going to ask this one to you.
Yeah.
I know what's going through my students' minds, and it's almost going through my mind. I'm a physicist. I'm a scientist, to be honest with you. And I thought I was a scientist until I tried learning this, and I realized I'm not very good at science. How do people actually use this? Like, I don't like learning things for the sake of learning things. Like, I like to see, well, okay, maybe I might not use this for the rest of my life, because apparently I didn't have to use this until now. But somebody did, and it's interesting to somebody. How is it used?
Well,
Why should I even care?
oh, okay. Well, what we're learning about now is protein synthesis. So your whole body is made out of proteins, right? And each of those proteins that we use comes from basically 27 different Lego pieces. And your DNA codes for those 27 different Lego pieces. So if ATG is a yellow Lego that's straight, right? That's one protein. And maybe CAG is a L-shaped protein that's red. If I change the codons to change from a yellow-shaped brick that I'm supposed to put into my protein to the L-shaped red brick, then I'll have a completely different structure, right? When I'm building myself up.
Okay, so what could people use that for?
So for us, we use it in like our hormones. Proteins are made out of hormones. So if I have, and hormones use lock and key shape. If we remember, like, did we go over cell structures and stuff?
Kind of. I don't know if I got into the lock and key.
Yeah, so the way our body works is if we have hormones, right? Our hormones are signals. If we want to feel happy, we send out happy hormones and our body accepts them. The happy hormone is shaped in a certain way that our body recognizes and latches onto and processes it. But if we have a mutation in our gene sequence, right, and our happy hormones turn into sad hormones, or they don't fit into that lock and key shape, our body doesn't work anymore.
So it sounds to me like this would be really good if you want to develop medicine that will help people.
Yep, medicines. It works for cancers because certain codons will tell you to start making proteins. What cancer is that switch, that on-off switch that a codon works for. It's broken somehow.
Oh, I see. Just something simple like saving lives. Huh?
Yeah.
Oh, okay.
Easy as that.
Sounds important. Okay, cool. No, thank you for. I always try to see the relevance in this stuff. You know what I mean? Oh, okay. All right. So, mutations. What happens if the guy copying the DNA screws up? Or what happens if the cooks don't read the cookbook correctly?
That turns into the Incredible Hulk.
Right, you get the Incredible Hulk. No, you don't, we're not talking about mutations as in like the X-Men, but maybe that could be a possibility, whatever. But we're talking about like when you're copying. All right, the 46 human chromosomes contain between 20,000 and 25,000 genes to be copied during DNA replication. Sometimes screw-ups happen. For example, one of the things you could have is the wrong letter put there. So this DNA, it should be an A, C, G, and A on the bottom, and it actually ended up being a, I don't know, oh no, an A, C, whatever, or A, C, A, and A, you know what I mean? And the T and A got all mixed up and whatnot. That could be a problem. Or what you could have is you could have insertion, where you have something like an extra,
I gotta go.
Yeah. Hey, thanks. By the way, Miss Yee, for all your help. You could have insertion, which is like an extra piece of an extra letter is added to the word or to your book. You're adding extra stuff. So this would be what happens if your thief sucks, if your mRNA, is junk, or maybe the mRNA, like was carrying the code in his pocket with an eraser, and half of it happened to get erased. These are different types of mutations. These are different types of screw-ups that could happen when copying the DNA. Well, here's what happens. I know you guys are thinking, oh, mutant, oh, that's basically when, like, you basically have, you can shoot laser beams out of your eyes then, huh? No, we haven't gotten to that mutation yet. But you could have cystic fibrosis, which is a defective gene where salt cannot move in and out of your body, and mucus basically builds up on the outside of your cells, and it blocks the lungs, and it affects digestion. It's a very sad disease. Or you could have Williams disease, or you can even have breast cancer or ovarian cancer. Very sad. Basically what's happening there is your body is not telling the protein factory to stop. I think with breast cancer, the proteins are just constantly being built, and you actually don't need it anymore, but your body doesn't know how to stop that process. That would be so. If you have these mutations, I think it's a bad thing. But apparently, not all mutations are bad. Some mutations don't have any effect at all. They just happen to, they happen to, like they might get messed up in the coding sequence, but maybe that screw up is a part of the 97% of the junk DNA that we didn't need in the first place. You know what I mean? So, hey, maybe that's why there's 97%, junk DNA out there. It kind of acts as like blank pages for you to, if those get messed up, it's fine.
Could that be like the cause of like disabled people?
Yeah, it could be a cause of disabled people. More so about genetic disorders. So we're talking about like cystic fibrosis and whatnot, something that you inherit, and that's not a good thing. Other mutations could be a good thing. For example, we know that your genes have something to do with your height. If you have a mutation in your gene, and all of a sudden this height thing gets all messed up, you could be like super tall.
Like 9-10 feet tall.
Yeah.
7 feet tall.
Yeah, you can be super tall. And a lot of people consider that a good thing, especially basketball players. So that's lesson 3, and we are done with chapter 5. I think that next week we're going to have a lab on this, and we're going to be moving into chapter 6. But I hope that, well, I'll be honest with you. I'm not going to be testing you too crazy on lesson 3, the one we just talked about. I did my absolute best to explain it to you. To be honest with you, I explained it to myself. I was like, well, this is what I think is going on. And then you guys were just so happen to be around to listen to me explain it to myself. But I'm doing the best I can. Hey, I just want to give you guys a heads up. If you guys are getting a D or an F inside of my class, I send emails to your parents. And I just made a new spreadsheet that allows me to do that really easy. So I'm just letting you know. I'm going to definitely be more on top of things, but I want to let you know so that way you can make sure that you guys are doing this thing. And then as far as the attendance goes, here's kind of how I do the attendance. I'm going to let you know right off the bat. Here's what goes on. I check to see if you access the class that day.
So all you need to do is like click on the class.
So all you need to do is really just go into the class. To be honest with you, between you and me, all you need to do is go into the class. You'll be all right for attendance. You don't even need to do any work. I would want you to do work, obviously, but you could just click on a link and you now have activity in the class. Wow, whatever. I check to see if you're in the class. If you did not access the class on the day that I'm taking attendance, then I check your grade. If your grade is above a D, so if you have a C, B, or an A, I still mark you as a 10, as good. The only reason why I would put an X on your attendance is if you both didn't attend the class or attend for that day, and you're getting a D or an F. Because in that case, then I'll say, well, then the student just didn't show up for that day. And if you get 3 X's in a row, then I send an email home to your parents. Okay?
Okay.
So if you go 3 days with both, we're going to finish in a minute. Give me a second. If you go 3 days with both not going into class and you have a D or an F, expect a email to your parents. And I happen to send it both to the student and the parent now, so, everyone's happy. All right? Any questions? No. You guys aren't in trouble. I'm just explaining to you guys what's going down. Want to make sure you know. I think it's only fair. Yeah. In fact, I'll be honest with you. I'm looking at the list. No one that is here is in real trouble. Well, never mind. I see one person. So, make sure you get your grade up, because I want you guys to do good. All right.
Okay.
Talk to you later.
Yeah, I gotta go too.
All right. Goodbye. That was fun.
Yeah.